Hello everyone and welcome back to yet another video from my channel Interactive Education, running for best education possible from the student to the student for a better experience. And today we're going to discuss and continue the unit we were discussing before, that is cell division and cell cycle. And today we're going to start with the second phase of the cell cycle, that is the M phase. So today we will be starting with the M phase of the cell cycle, which stands for the mitotic phase. Mitotic phase. Basically, this phase deals with the actual cell division. So in the interphase, we only discuss about the preparation for the division. We don't actually go into the division process as such. But in the M phase, we always talk about the actual cell division, as this is the phase in which we have the mitosis process taking place. However, it is to be noted that meiosis can also take place. However, I will not be discussing meiosis. I will not be discussing meiosis because it is a bit too complex. I will be taking it up in another video maybe, but for now my main focus would be the mitosis process only. So let's get started without any further ado on to our topic for today. So if you look at the previous video, we discussed about the interphase. And we saw how the interphase is basically involved in cell division preparation. So we basically prepare for the process of cell division, but we don't actually talk about the process as such. But in M phase, that's what's actually taking place. So just like the interphase had three subphases, the M phase too has some subphases. It has mainly two subphases, or better not call it phase, but call it a stage. So there are two stages of M phase. So stages of M phase. There are basically two stages of M phase. Firstly, we go for the karyokinesis. And the second stage is the cytokinesis. Now, karyokinesis is a word which karyo is a word which is associated to nucleus. So karyokinesis basically means nuclear division. Division of the nucleus, because obviously every daughter cell needs a nucleus. It can't exist without that. So the nucleus needs to divide first into two, and then we have the cytoplasm which divides. So cytoplasmic division. So there are two phases of M phase. We have the nuclear division phase, which is the karyokinesis, and then we have the cytokinesis phase, right? Now let's talk about each of the phases in detail. Firstly, we have the karyokinesis, which is the most complex procedure and the longest phase of the M phase. And we know that the M phase itself only constitutes about 5% of the cell cycle only. But if we say in the M phase specifically, then we have karyokinesis, which is the longest phase in the M phase. So karyokinesis basically is nuclear division. And in nuclear division, again, there are many other stages. So karyokinesis again is further divided into 4 phases. So again, karyokinesis 2 is a complex procedure. Karyokinesis has 4 phases. We have the prophase, we have the metaphase, and finally we have, excuse me, not the metaphase, anaphase, and then telophase. So we have 4 phases of karyokinesis: prophase, metaphase, anaphase, and telophase. If you see some books, textbooks, and even websites cite another stage which is a pro-metaphase. This comes in when you go for cellular study, because it is again a very in-depth step of karyokinesis, which I will not be discussing here because it is a part of research. So I will not be discussing it here. I will be discussing the main 4 phases: prophase, metaphase, anaphase, and telophase. Now this karyokinesis process basically takes place in these 4 phases, and let me tell you, today I'm going to have a hard time teaching this because I have to draw many diagrams. Prophase. What's wrong with my stylus? Yep, okay. Prophase. So this is the first phase of karyokinesis, prophase. Now we have seen this before, that the cell is like this, and it has a nucleus, right? Now, if you just have a glance at this nucleus in detail, if you open this nucleus up, which we obviously cannot, but just assume that you go into the world, you're in an imaginary world, and you can just open this nucleus up. So when you open this nucleus up, you will see something like this, right? So you're going to see this as the cell, as the nuclear membrane. Then you're going to see a, dark kind of structure just inside. You're going to see some winded up threads, right? Some threads which I have just, wounded together. And you also see some degree of ribosomes. Right? So this thread-like structure and this rounded, winded thread kind of thing is nothing but the chromatin material, which is the material which contains the genetic material. It is the, the brain kind of, mainly the storage place of the chromatin. And this dark kind of structure, as you know, is the nucleolus. So this was just a glance at the nucleus, and this membrane is the nuclear membrane. It is also called, since we know that karyo is a word related to the nucleus, and we know that the cell membrane is also called plasma lemma. So the nuclear membrane is also called karyolemma. Karyolemma. That is the covering of the nucleus. So this is basically a distribution of how the nucleus is. But let's just now get back to our topic. We saw all this, right? The internal structure of the nucleus. And now we need to look at the prophase stage. The stage of prophase basically involves the disintegration of the nuclear membrane. That's one of the major things which take place in the prophase. That there's a nuclear membrane. This nuclear membrane starts or begins to disintegrate. So the first thing which takes place is disintegration of karyolemma. So the nuclear membrane actually starts disappearing. It's just going away. It's flashing off from the world, from the cell. Disintegration, so going away. So this nuclear membrane starts disintegrating, and the next important thing which takes place is the condensing or the condensation of chromatin to form chromosomes. Right, condensation of the chromatin to form chromosomes. Right, so these are the two main things which take place in prophase. Basically, we're trying to now, since we're dividing the dividing the nucleus, now we have to make a new membrane, and also we're just disintegrating the nuclear membrane, and we're also condensing the chromatin. And just recall from the interphase that in the interphase we saw that chromosomes double up. And I had taught you this in the interphase when I was teaching interphase, and this takes place in the S phase of the cell cycle. I hope you remember that, and it's better you go over that video if you have not. So chromosomes have doubled, and whatever chromosomes we have now, like we have the 4N chromosomes as compared to the 2N before, these 4N now which are stored in the chromatin material now start condensing to form chromosomes, right? So these are some of the main things which take place, and you see that this will be somewhat the diagram that will actually just will show you what's happening in the cell at the time. So suppose this is my cell and this is my nucleus. I had shown you how the nucleus looked like before. It was completely, a very well built dot kind of structure, right? So now you can see that the nuclear membrane has started disintegrating. You can see that it's becoming loose. This is why I'm showing it with a dotted line. And again, the nucleolus which is present, it also starts disappearing. Now, there is no more nucleolus. The nucleolus has disappeared, just like this nucleus, right? And now, with this too, the chromatin start changing to chromosomes. So somewhat like this. They start forming some chromosomes. Right? I hope that's clear to everyone. Now, that's the prophase. Let's come to the next phase of the cell cycle. The next phase of the cell cycle is the metaphase. So we looked at how in the prophase the chromatin is condensing to chromosomes, the nuclear membrane is breaking, it's disappearing. But if you go into the metaphase, the nuclear membrane has completely disappeared. Nuclear membrane completely disappears. Now you have no more nuclear membrane left. So, oh, my nuclear membrane has gone, it's disappeared. So it has vanished into the cell. The second thing which takes place during the metaphase, after the nuclear membrane has gone, is chromosomes complete. Now, I've completely formed the chromosomes, right? The chromosomes are complete. I have got completely condensed, beautiful chromosomes. Now, let's have a diagram to see what's actually happening. Now, that suppose this is a cell. Now, in the cell, you saw here that the nuclear membrane has disintegrated. But let's just look at one more thing. You know that there's a special cell organelle which plays a major role in cell division. That is called the centriole. That's the centriole. So centriole plays a major role in cell division. Let's see how now. So there's one thing that during the G2 phase of the cell cycle, of the interphase, the centrioles duplicate. So the centriole duplicates. It forms a duplicate of itself to give to the other cell in the G2 phase. So centriole duplicate, and you will see that the centriole during the metaphase each centriole goes to each end of the cell. So one centriole on each end of the cell during the metaphase. And now as we saw earlier, the chromosomes have come out, they've condensed, and the nuclear membrane is completely gone, right? That's what happened. Right? But there's one more thing which is very, very important and takes place here. That is the centrioles begin releasing spindle fibers. Now the centrioles begin releasing spindle fibers, and have a look how these spindle fibers look like. It's like this. So they begin releasing like they are Spider-Man, right? They're the Spider-Man. The centrioles are the Spider-Man of the cell. They begin releasing their webs, right? They begin releasing their webs, and you will see that ultimately at the end of metaphase, this is how your cell is actually going to look. You're gonna have the nuclear membrane. Sorry, you're gonna have the cell. You're gonna have the centrioles, and ultimately, as we looked at the spindle fibers earlier, they're gonna be like this. So we're going to have something like a globe, right, like a small earth, and in the earth we have all these latitudes, right? They're like the latitudes and the longitudes of the earth, which have been, released by the centrioles. And where in the world are the chromosomes? Well, the chromosomes are going to attach themselves to a particular point, and one chromosome to each of the spindle fibers. And how in the world are they going to attach? Well, if you look at a chromosome closely, you will see that there's a point, right, where this chromosome and this chromosome, they have met, right? Because you have this and this which come together. So there's a point where these chromosomes meet, right? And this point of, touching, you may say, or a point where it's like that, the center part of the chromosome is called the centromere. And each spindle fiber which is being released by each of the centrioles, they too, there's a spindle being released by this centriole and a spindle being released by this centriole, they come and meet at a point. And that point of intersection or meeting of the spindle fibers released by each of the centrioles is called the kinetochore. So that's the kinetochore where these two are meeting. So basically what the chromosomes do is that they go and attach to the kinetochore with their centromeres. So their centromeres and the kinetochore attach to each other. So if you see, that's how they're going to attach to the spindle fibers. And that's what happens in metaphase. That's the actual thing taking place in metaphase. The third phase of the cell nuclear division process is anaphase. And anaphase is, again, okay, let me use blue for the cell now. Oh, my friend yellow is okay again. So that's my cell again. Now we looked at the cell before. You can see here that all the chromosomes that have attached themselves to each spindle fiber. Keep in mind here that the total number of chromosomes is still 4n, as it duplicated during the S phase in the interphase, so it's still 4n. But now these centrioles, which have created these webs like Spider-Man, so these centrioles which have created these webs, I will be making only a few now, not all of them. So these are my chromosomes, with their attached kinetochore and centromeres. And basically these chromosomes which have attached to the kinetochore with their centromeres, basically now this chromosome, right, you can see that we make it like this. This is one chromosome and there's another chromosome, they meet and they form a pair of chromosomes. So this chromosome here, you can see this one, is being pulled by this part. So this chromosome is being pulled by this part, whereas, let me denote it with another color, this chromosome here, right, this one, this chromosome here is being pulled by this spindle fiber. So this chromosome pulls this spindle fiber and this chromosome pulls this spindle fiber. Same goes for all the other chromosomes, right? They are being pulled by their respective spindle fibers. They are pulling it here, 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 and here. And the yellow, you can see that this one is being pulled here, this one is being pulled here, and this one is being pulled here. So basically, the chromosomes are being pulled to the two ends of the cell. That's what's happening in anaphase. And by the end of anaphase, we see that this is what the case is. These are the two centrioles, right? And they're releasing their webs of doom for the chromosomes, and the webs of doom, the webs which are here to separate the two sisters. I call them sisters, and they are actually called sister chromosomes or sister chromatids. So this is one chromatid, this is another chromatid, and these two are called the sister chromatids. And by the end of this phase, this is what the case is. We have separated successfully the two sister chromatids from each of the centrioles. So you will see we have successfully separated them. This is one sister chromatid, and if I take my color back, or this one, we'll see that this, and this, they have been successfully pulled by each chromatid, by each spindle fiber. So this is what happens. These two sister chromatids have been pulled apart. So this happens in such a way that one part contains two N chromosomes, while the other part contains two N chromosomes. I hope that's understood. Now we have divided them in such a way that each part contains two N chromosomes, and we saw here that the total is still 4N, but now it has now divided. Right, so that's what's taking place in anaphase, the separation of the two sister chromatids, a short phase, I would say. And finally, lastly, we go on to the last phase, which is your telophase. Now, what's wrong with my stylus? So now we move on to the last phase, which is a telophase. And again, let me pick up my favorite guy, that's the yellow. So we come here and we are back with another cell. But this would not be correct to be drawn right now. We should draw something like this. Right, that's my cell. Why would someone would ask me, when you're drawing something circular here, why drawing something oval or potato shaped here? It's because we have our great nuclei back. Each of the two centrioles pulls the one sister chromatid from each spindle fiber towards itself. So by the end of this, you will see that the centriole is here, it is being pulled, and there's one chromosome here. Right? So, or better use that color. Right? And you will see that there are various chromosomes now which have come together. And they again form their chromosomes, right? Similarly, if you look at the other side, it's similar. They have their own chromatids, and they now have formed individual sister chromosomes, because here, when they're being pulled, these original parts are now called chromosomes, single chromosomes. When they have been pulled, they are now called single chromosomes, and that's what happened here. All the chromosomes went and they combined with their nearest relatives and they formed something like this. What happens after this is that now each of the two starts forming their own nucleus. So if you look here, you have the nucleolus appearing here back, and these chromosomes now start decondensing back to the chromatid material, and they form one individual nucleus. The same goes for the other. a separate nucleus is formed, a nucleolus starts forming, and you have your own chromatin material because the chromosomes decondense. Like, we know the chromatin material condenses to form chromosomes, but here the chromosomes have decondensed to form chromatin. And ultimately, the final part of this process of karyokinesis will be an elongated cell with 2 nuclei. Right, that's the end product of karyokinesis, a 2-nucleated cell. And after this, immediately follows the cytokinesis process. Right, in case of an animal cell, so now after this process, which is common to both plants and animals, we move on to, this is how your karyokinesis is taking place. And now we move on to the second stage of M phase, which is called the cytokinesis process. So we looked at this, the karyokinesis. You saw that how in the end, after all this complex procedure, we come back to a cell, or we come to a cell which has 2 nuclei, but now it's still one cell. We need to break it down into 2 daughter cells. So here what we do, basically we're going to discuss 2 cases, animal cells first of all. In animal cells we have only one, we know that we have only one simple, this guy, simple plasma membrane. So this multinucleated structure looks like a guy's eyes, or an alien without a nose and mouth. So that's what's happening. Here you have this cell, elongated cell. Now after the cytokinesis process, or during the cytokinesis process, this starts dividing. So you start getting 2 cells with separate nuclei. So this is what somewhat the thing comes out to be. Now that they have started now dividing. A barrier starts forming. They start dividing because you see there's a barrier in between them. We have to separate. We are India and Pakistan. So they start dividing. Alright. So each one has its own nucleus, and ultimately you will have 2 daughter cells. That's what is going to take place. You're going to have 2 nuclei and 2 daughter cells. Two daughter cells. Right? That's what's going to take place in this process. Right? So that's how it takes place, or that's how cytokinesis or cytokinesis takes place in the animal cell. You have you have this guy which has 2 nuclei. This 2 nuclei guy breaks its cytoplasm and forms 2 daughter cells with separate work, right? And now one important thing to note, since it is mitosis, each cell will have 2 N chromosomes. And as we showed here, it is very, very easy, and that's a very complex procedure, how we got them both to have 2 N chromosomes, right? So that's with respect to your animal cells. If you go to the plant cell, it's a slightly more complex procedure. So now we move on to the plant cells. If you go into the plant cells, it is slightly more, a bit more different. Basically, in a plant cell, we know that, okay, the karyokinesis is going to be the same. Okay, you're going to have a cell, okay, which is going to get 2 nuclei. But remember that this guy, the plant cell, has a cell wall, which is rigid. It is not easy for it to do this, right? But this cytokinesis process is slightly more complex because there is the presence of rigid cell wall. So because of this, it becomes slightly more complex. But I will be making a separate video on cytokinesis in plant cells. So I will be doing this in a separate video just after this, right? I hope you'll get your answers there. So because of the presence of rigid cell wall, it is not as easy in animal cells. There's a slightly more complex procedure, right? So I'll be taking that up in the next video. So with this, we conclude this video. I would like to finish this off and wrap it up here. Thank you very much for joining me today. I hope you enjoyed. I hope you enjoyed my diagrams, and I tried my best to make them best. Please let me know if you have any suggestions in the comments section below. Thank you very much for joining me today. Stay healthy, stay smart, and if you have any doubts, please, I welcome them in the comment section below. Goodbye, everyone. Goodbye. Stay healthy, stay smart, and keep studying every day.